Hey, it's Horner, and let's talk a little bit about DNA prep and gel electrophoresis. So remember, the first thing we have to do is we have to get the DNA out of the cell. So when we did the strawberries, we put it in a solution that had salt. And what happens is, because there's salt there, these cells swell up really big, and then as you squeeze them, they pop. And that makes all of that stuff come out. Then we put a little bit of ethyl alcohol in there, and remember, that separated it out. Once you do that, there's some process called polymerase chain reaction. And all that does is just make a ton of copies of all of the DNA. We want a lot of copies because when we go to put it in a gel, we want to make sure that there's enough there that it shows up or it glows inside that gel. So the next step after we have all the DNA that we need, and then maybe even a little bit extra, we have to cut it. Remember, that's called restriction digestion. So we're cutting the DNA at specific places using enzymes. And each section that we have has a certain number of base pairs. So let's take a look at some of these restriction enzymes that we can use to cut. So there's one called EcoR1, one called HAE3, and one called HIN3. And the one that we used in class was the one that was HAE3, and that cut between a GGCC on the top strand of the DNA. And you'll see that if you ran the other way from right to left, it would be GGCC on the bottom strand, but not CCGG. So anyway, if you do that, remember, because it's a straight cut, we call that a blunt end. And if we cut it and we use EcoR1 or HIN3, you'll notice that it's got this jagged shape. And remember, we call that a sticky end. So this one is a sticky end because it's not straight cut through. And then this other one would be called a blunt end, because that's essentially what you've made, is you've made a blunt cut, or we term this top one a sticky cut. The sticky cut is the same thing as we also have a sticky cut down here on the bottom. So this one also produces a sticky cut. You won't need to memorize what these are in terms of sticky ends or where they do their cutting. You just need to know how to use them. So let's go ahead and take a look at an example. Let's say that we want to cut this one at HAA3. So we'll say that that's our restriction enzyme. And it's going to cut between GG and CC. So for this first one, we'll just go ahead and we want to put a cut mark right between the GG and the CC. And then we're going to just keep going to the right. Oh, here's another GGCC pair. And then we're going to come all the way over here, GGCC again. And we've made 3 cuts. So when it says how many cuts, you just have to put down how many times you were able to cut that strand of DNA. They want to know how many base pairs are there. So we need to count the base pairs. Now, base pairs, remember, are any time you have a top and bottom pair. That's a base pair. So right here, we would have a base pair. So there's 1, and then there's 2. So this one has 2 base pairs. So we would just put a 2 here. The next one has 3, 6. So this one has 6 base pairs. We're just going to count along the top again. There's 5. 6, 7, 8, 9, 10. So this one has 10 base pairs. And we have one more section, and this section has 8 base pairs. So now we've got 2, 6, 10, and 8. So what are the base pairs? They are 2, 6, 8, and 10. Now that we've cut the DNA, we need to go through and do the last thing. And that's to put them in a gel and run them in that gel so that we can see what our sample looks like. So we're hoping that got cut on the right places. And if you remember, in gel electrophoresis, we put a sample inside So here is the ladder that has all of the base pairs. Where we have person 1, remember we had person 1 here, and we said that these are the number of base pairs in each one of those sections. So when we put the DNA on the negative end, then turn on the electricity, that electricity will then, because DNA is negative, so the DNA inside here is negative, it wants to run towards the positive end. And it does not like the negative end at all, so it's trying to get away from it. When we did the sample, you'll see that the heavier DNA parts, or that the one with the most base pairs, which is 18 for the ladder, you'll see that it came out there. Then you had a 16, you had a 13, a 12, maybe a 9 or so, maybe a 10, an 8, a 6, a 4, and a 2. For our person, we had a 2, a 6, an 8, and a 10. So for the person we have, these moved so that they are at 10, 8, 6, and 2. And that's because that's what we cut them at using the restriction enzyme. And that is all there is to it.